So our headquarters are based in Craigavon in the UK. We also have a clean testing laboratory in the US, which is based in North Carolina. And we have recently established a partnership with WuXi Nextcode, and this was specifically set up to enable us to deliver a clinical trial to the Chinese market. And this was necessary because, as most of you will know, it's not possible to ship samples either in or out of China. So in order to get this set up, we sent a team from Almac out to China in order to train the WuXi site and tech transfer in the clinical trial assay that we had developed in the UK. We then oversaw the validation required for the delivery of this clinical trial to the Chinese market. So having given you a bit of background to us as a company, I wanted to move on to discuss the benefits and challenges of NGS panels for subject selection. So the obvious benefits of NGS panels for subject selection are the fact that NGS is suitable for both DNA and RNA samples. So for DNA, you can be looking at single nucleotide variants, insertions and deletions, and copy number variation. And then with RNA, you have the ability to look at gene fusions and splice variants in addition to gene expression. But I suppose the real strength of next-generation sequencing is the capacity to multiplex on a huge scale. And with this ability comes the ability to capture really complex biology. And it's for this reason that next-generation sequencing is considered a really powerful tool for biomarker discovery. But potentially the ultimate benefit of NGS is the potential to report multiple biomarkers from a single sample and a single wet lab process. And I suppose if you can harness this potential, there's the ability to maximize the utility of precious patient samples and provide more information to direct treatment decisions. However, with these benefits, there are associated challenges, and it's important that these factors are taken into account when you're using NGS platforms for the development of diagnostic tests. So it's really important to carefully consider the design of your broad panel, and also to make sure that you have a clear justification for the intended use of the panel. So for example, are you wanting to have a panel that will cover multiple biomarkers? And if so, what settings do you intend to use this panel? Or alternatively, do you simply want your next generation sequencing panel to be used for a tool for biomarker discovery? And really, depending on your answers to these questions, there are cost and throughput implications. So it's very important to have a clear objective going into the design of any of these studies. And the final thing, I suppose, is that to be conscious that with the additional complexity that you add, it's important to be aware that this could lead to additional challenges for analytical validation and also the regulatory pathway which you may wish to pursue. So in order to look at this in a little more detail, I would like to review a few examples of case studies of NGS panels that we have run at Almac Diagnostics. I'll begin then by looking at an off-the-shelf panel which we are using as a research-use only screening tool. This work we've performed in conjunction with EORTC, and for those of you that aren't familiar, this stands for the European Organisation for the Research and Treatment of Cancer. And EORTC established SPECTA, which stands for the Screening of Patients for Efficient Clinical Trial Access. This SPECTA consortium is a pan-European multidisciplinary network, and it's made up of representatives from academia, industry and healthcare providers. And they have adopted a centralised model to deliver precision medicine, and the overall aim is to try to streamline patient inclusion in clinical trials. To give a little background, this is summarised in the model here, in that patients are submitted to a centralised biobank for biomarker screening. Once the biomarkers have been screened, all of the data is returned to the centralised database, and then it can be utilised for both biomarker discovery and potential diagnostic development by all members of the consortium. And really, the overall drive behind SPECTA is to have a focus on quality assurance and quality control. And it's really for this reason that they've adopted a centralised model. So they have centralised pathology review of the biobank, and then they also have centralised model for molecular testing. And this is really where Almac Diagnostics comes in, because we're the preferred partner for the molecular profiling of EORTC clinical trials, including that of SPECTA. In relation to the work that we're actually running for them, we are currently running the Illumina TrueSight Tumour 170 panel on retrospective SPECTA cohorts. For those of you that aren't familiar with the panel, this was developed for use in formalin-fixed and paraffin-embedded tissue. So whenever a sample comes in, samples are subject to pathology review, and then RNA and DNA is extracted and quantified from each sample. This is followed by hybrid capture and library prep, followed by sequencing on one of the Illumina platforms. So this can be processed using either the NextSeq or the HiSeq platforms. In relation to the workflow, this process enables the parallel processing of RNA and DNA derived from FFPE solid tumours. And the RNA and DNA combine at the library prep stage. So RNA and DNA samples go through exactly the same workflow following the cDNA synthesis stage or the sharing of genomic DNA, and this is just summarised on the right-hand panel. In relation to what this next generation sequencing panel covers, it covers all coding exons of 170 genes, and it also includes a range of variant classes which are common in solid tumours. So you can see again, you won't be able to get resolution on this, but the table on the left-hand side shows that the TST170 panel covers 151 single nucleotide variants, or insertions and deletions, along with 59 amplification events. And from the RNA component of the sample, it covers 55 fusions or splice variants. So Almac have a strategic partnership with Illumina, and it's through this that we were initially involved in the beta testing of the TST170 panel before it came to market.